DNA analysis was performed at the Hubbard Genome Center at the University of New Hampshire and was funded by New Hampshire Embry. So thank you to New Hampshire Embry for funding this project. My name is Autumn Burlide, and my project was on the microbial communities present in natural snow. For this project, we wanted to explore the microbial communities that are present in natural snow from a farmhouse in Charlestown, New Hampshire. The snow was also exposed to farm equipment. It's important to note that in this experiment we used natural snow and not man-made snow, which is often common in ski resorts. According to research, Pseudomonas syringae is the most popular microbe in snow. We collected 2 gallons of snow and stored it in negative 20 degrees Celsius, and then we got to work on our agar plates. Pseudomonas typically grows best on Kingsby agar, so we used Kingsby agar to make our plates, and the recipe is seen below, and it was provided by Tim Hastings. Once we had collected our snow and made our plates, we removed the snow bags from the freezer and warmed them up until they were liquefied. Once they were liquefied, we plated the snow using the 4 quadrants technique, and then we stored the plates in the 30 degrees Celsius incubator. Once the colonies formed on the plates, we removed a small biomass and transferred it into a sterile tube to be sent out for DNA analysis using Patrick. For more information on Patrick, you can see our works cited and the following slides. Shortly after sending out our sample, we received results on the taxonomic analysis. My sample was 86.58% gamma proteobacteria, and of that, 78.5% was Pseudomonas, which are gram-negative rods that can cause critical infections such as endocarditis, meningitis, and pneumonia. Typically, these microbes are found in water, dirt, and plants. Here is a breakdown of the percent Pseudomonas, and you can see that the most common Pseudomonas is Pseudomonas fluorescens, and then unclassified Pseudomonas, and very small amounts of several other types of Pseudomonas. Pseudomonas are motile bipolar flagella, and sometimes they actually have more than one flagella. In this picture, you can actually see that the pseudomonad has several flagella. Typically, Pseudomonas is not pathogenic to people, and for it to be pathogenic, it would actually have to be introduced via the blood. So, for example, during an operation where a dirty tool was used, or intravenously during drug use, or some sort of medical operation. It's actually very interesting because it was believed to be a causative agent for patients with pulmonary illnesses and pulmonary diseases because it was found in aspiration samples from these patients that had these pulmonary illnesses. But it was found to be in the water of the humidifiers that these patients were using, and it was not causing their illness, it was just in their environment, which goes back to Pseudomonas being very common in water, dirt, and plants. I had a relatively pure culture, but it still was not 100% pure. There were several different types of Pseudomonas within my sample. But the colony morphology was flat, smooth colonies that were only a few millimeters in diameter with regular margins, meaning they were not oddly shaped, they were typically circular. Next, we did sample staining, and I did a simple crystal violet staining, and with this crystal violet staining, we observed purple rods, and pseudomonads are typically 1.5 to 3 micromillimeters long and 0.5 to 0.8 micromillimeters wide, which is consistent with this image at 1,000 times magnification. Pseudomonas gets into the snow and dirt and plants, often by the wind, and they're able to land on these things because they're encouraged to form ice via ice nucleation. And This aids in the formation of clouds that lead to contaminated snow. For a more in-depth explanation of this process, I recommend checking out the scientific article that I have linked down below by the American Society for Microbiology, and it delves more into the topic of ice nucleation within Pseudomonads. Our classmate Ashley actually found very similar results with her sampling of lake water, which shouldn't be too surprising given that Pseudomonads are typically in water, snow, soil, and plants. Her results were 30% Pseudomonas fluorescens, which is not far off from how much of a percentage mine was of Pseudomonas fluorescens. So that is a very interesting find that I found. Here I have listed my sources. I highly recommend checking them out if you have any further questions. If not, you can email me or leave a comment. I am very interested in seeing what you guys have to say about this. And I was very, very happy to see that my results were within reason of what I expected. I wasn't specifically expecting to see Pseudomonas fluorescens, but it was very nice to see that I at least had Pseudomonas in my sample. So thank you guys so much for listening to this video presentation, and thank you for an amazing semester. This has been a really interesting class, and I'm so grateful that I got to connect with so many great peers of mine, and I hope you guys have a great summer. Bye.